Hello, and thank you for attending this talk. So my name is Wade Elliott. I am a research chemist at the National Institute of Standards and Technology, and I'm going to be presenting some data on some of the current methods and metrics for chemometric analysis of NMR spectroscopy methodologies for analyzing monoclonal antibody therapeutics in their higher order structure. So to give a very brief outline of what I'm going to be covering in the next 20 to 30 minutes, I'm going to discuss a little bit about monoclonal antibodies and more importantly, why we care about being able to analyze their higher order structure and why we care about the chemometric aspect of that. I'm going to dive a little bit into a couple of different methods that have been published and some of the studies we've been performing along with those methods to show the robustness and usefulness of the techniques. And then I'm going to present a little bit on the utilization of lower field, lower resolution NMR spectroscopy as a modality for screening protein therapeutics. So monoclonal antibodies currently command a very large portion of the pharmaceutical revenue stream. As you can see here, this slide from the sales of the top 10 selling drugs in 2017, the overwhelming majority of these are monoclonal antibodies or protein therapeutics. And so as you can tell, with this much money being poured into research and development, it's very important that we have well-established methodologies in order to be able to analyze these therapies. Furthermore, as the years progress from the time that these drugs are developed, we get closer and closer to the time in which these monoclonal antibody therapies and other protein therapies are going to come off patent. This is going to make way for a huge wave in the biosimilar production realm. Why that's important is the way that we evaluate and determine the usefulness and safety and efficacy of these protein therapeutics is inverse between the originators and the biosimilars. So when we deal with originators, typically we do some developmental analytical work. We then proceed into some non-clinical based tests and trials, and then we move into clinical trial where we determine the potency, the efficacy, as well as the safety. However, with respect to biosimilars, we do the opposite paradigm, where we spend most of our time analyzing the actual molecular species themselves to make sure that they are in fact similar. And then we do a little bit less into the non-clinical and clinical methodologies with the understanding that as long as the structure is within a specific realm of similarity, that realistically the functionality should be the same. So why is NMR spectroscopy useful with respect to protein therapeutics? Well, NMR is unique in that we can get very, very specific information with respect to individual atoms along with individual amino acids in the protein and how they're interacting with each other. So one problem that is presented, though, with respect to monoclonal antibodies versus a small peptide or a small protein, such as ubiquitin or GB1, is that MAbs are on the very, very large end of the spectrum by comparison. They averaged around 150 kilodaltons, and because of that, the motion of these proteins in solution is very slow. And that's something that is disadvantageous with respect to NMR. So as you can see here, there is a spectrum of a proton and nitrogen correlation. This allows us to look specifically at that amide region of the backbone of the protein. One of the problems with NMR, besides the necessary or the need for fast motion in solution, in this case, is the low rate of NMR active naturally abundant isotopes. So protons, for example, exist at nearly 100% natural abundance, which allows us to take full advantage of all of the hydrogen atoms, or at least the overwhelming majority of hydrogen atoms in in our sample. Conversely, nitrogen-15 in this case exists at a less than 1% natural abundance, which means that for every 100 for every 100 nitrogen atoms that are in the protein, we are only seeing about 0.5 of them, or 0.3 actually, 0.37. So that makes it much less sensitive than some of the other techniques out there. Now, we can get around this by doing things like isotopic enrichment, but when we're dealing with drug products, that is usually something that is not regularly done, which is isotopic enrichment of the protein, which means that we have to find other ways to be able to get NMR data, specifically things like these NMR fingerprints that I'm showing here. So there was, a few years ago, a method that was presented called Profile, which stands for protein fingerprinting based on line shape enhancement. The idea is that you utilize proton NMR, which has a very high resolution, very high sensitivity, and you have a very high natural abundance of protons in your sample, and you can get these very detailed spectra. In this case, with this profile experiment, we're collecting a pulse field gradient stimulated echo type of experiment, which is a fancy type of diffusion experiment, which allows us to get rid of any of the excipients and suppress the water signal out, so all we see is the protein. What we then do with this data in the profile method is we apply a Gaussian line broadening function to produce this contour spectrum that you see. We then do some subtraction of the original, of the contour spectrum from the original spectrum, and we're able to produce this fingerprint. What we can do from there is take it a step further, and we can take this fingerprint spectra and compare it to the fingerprint spectra of other protein therapeutics or protein therapeutics from the same lot. So we can get an idea of similarity or dissimilarity. Now, a few years ago, a couple years after the profile paper was published, our group here at NIST published a paper on using methyl groups as a method of fingerprinting things like higher order structure and fingerprinting antibodies, specifically these monoclonal antibodies. The idea with this is that from a natural abundance perspective, carbon-13, which is an NMR active isotope, exists at about a 3 times rate to that of nitrogen-15. This means it's a much more sensitive technique than the previous, or the couple previous slides I showed where I showed the nitrogen or proton nitrogen HSQC. This allows us to get more detailed information. And in addition to that, as you can see on the picture on the right, the methyl groups are very evenly dispersed throughout the entirety of the antibody, which gives us information about localized higher order structure changes from the secondary structure, tertiary structure, and even potentially the quaternary structure and quaternary structure, depending on what kind of interactions and changes we see. So we were able to collect very detailed data. The first spectra you're seeing here is at 900 megahertz, which is a very high field NMR. And we can even go at slightly lower field at 600 NMR, or 600 megahertz, which is also still a high field NMR. Okay, so what we can do with this data is we can collect these spectra and compare them and generate similarity information based on types of correlations. In this case, I'm showing a linear correlation analysis between 2 different antibodies, which the spectra we see up in the upper left-hand corner. We can then bin this data and do a direct correlation comparison, and in this case, get an R statistical value to give us a relative similarity. So we took these 2 methods and we decided to run a multi-lab study in order to determine if these 2 methodologies, which have been published, are in fact comparable. And so as you can see here, there's a list of numerous blends of antibodies along with antibody types in which we can go through and actually do some comparability studies between the 1D method and the 2D method. Now, all of these spectra were collected on the list of NMR spectrometers that you see at the bottom. And we were able to take that data and do some interesting things and find out some interesting things, which I'm going to start showing you in just a couple slides. So here we have a list of the correlations that we performed after we collected the data. And as you can see, it's a symmetrical correlation, which allows us to skip a little bit of extra work. What we did as far as the experimental details are concerned, with the 1D experiments, as I stated previously, we run a pulse field gradient stimulated echo experiment. Each of these experiments take approximately 2 hours, actually around an hour and 30 minutes to complete. We collected 5 data sets per sample. And then we processed initially the data in topspin using some Gaussian line broadening, the automatic phasing, as well as taking it a step further and applying the profile processing scripts in MATLAB. With respect to the 2D data, we collected gradient HSQCs with water suppression. Each of these experiments took about 2.5 hours, and we collected 2 data sets per sample. The processing was then performed in NMR pipe with cosine bell squared apodization. And since these experiments typically take fairly long to do, we acquired at 50% non-uniform sampling. So we used IST reconstruction and some automatic phasing, and then did cross-correlation analysis in MATLAB. So what I'm going to show you here is some of the profile data for the 1D correlations that we did. What this is representing is the first 4 samples. So sample 1 being IgG1A, or sample 1A. Sample 2 is an IgG1, which is a sample B. Sample 3 is the digest of sample 1, and sample 4 is the digest of sample 2. And what you can see is the correlation from each of the spectrometers is actually very, very similar to the others. So the trend is very similar between each of the spectrometers. And that gives us good information that this technique is, with respect to these samples at least, very robust. And just to illustrate the point a little bit more, these are the correlations on a bar graph. So you can see that the relative correlations that are calculated are actually very, very similar across the different spectroscopic fields. For the 2D data, we performed, as I said, a linear correlation in MATLAB. And you may notice that some of the data is a little bit different. I'm going to go into that in just a few seconds. The samples we're looking at are the same samples that we looked at with respect to the 1D data. And you will also see that the 2D data is very comparable within its own subset of 2D data. And once again, the bar graph illustrates that each of the spectrometers gives you fairly similar data. But as I mentioned, you do see that the trend between the 1D profile method and the 2D cross-correlation method are slightly different. And I'm just going to highlight a couple of instances. Now, the circles that I'm highlighting of these box and whisker plots are illustrating, specifically in this case, the comparison between the IgG1A and the IgG1A digest. Now, the reason we're seeing this discrepancy, not just with respect to this one, but some of the other discrepancies in there, has to do with the size differential. And I mentioned earlier that size with respect to NMR spectroscopy is fairly important. So if we look at the 2D spectra of sample 1, which is the IgG1A, and sample 2, which is the digest, we can see that they don't match. So the digest is the fuchsia colored, and the gray is the IgG1, or the sample 1. As you can see, there are peaks that show up with respect to the digest and do not show up with respect to the intact product. The reason for that has to do with what is known as chemical shift anisotropy. So larger molecules rotate or tumble at a much slower rate. And because of that, we experience a significant amount of line broadening. So in order to compensate for that, we typically need to acquire our data for longer or take more scans of our data. This was actually illustrated a few years back in a paper produced out of our group as well, that doing or performing a domain cleavage, meaning doing a digest of the intact MAb into its FAB and FC domains, does not actually alter the higher order structure, but we just require more scans of the intact MAb in order to be able to dig down deeper and account for the line broadening. So we did additional correlations outside of just those 4 samples I showed you. And as you can see, once again, profile for the most part across the spectrometers gives you fairly comparable data. With respect to the 700 megahertz spectrometer data, you will see that there is some discrepancy compared to the other 3. And that is due mostly in part to an issue that was an issue with water suppression of the sample setup or the experimental setup. So that is the reason that data doesn't necessarily match. If we go over to the 2D methyl fingerprinting data of the same sample correlations, we begin to see a very significant discrepancy between or across all of the spectrometers, with the exception of the 600 megahertz A and 900 megahertz spectrometer. The reason for that, well, and I'll get into that in a minute. I want to show you the data first. So sample 12 data, or excuse me, sample 12, which is a blended sample. This is the spectra of the sample taken at 900 megahertz. And I will point out that the 900 megahertz spectrometer data is actually some of the very first data that was collected. If we look at it compared to the 600 megahertz spectrometer data, you will see that there are some significant discrepancies with the data. And that has to do with the fact that the 600 megahertz 600 megahertz B data was actually collected at the very end of the study, which was close to a year later. We see that with respect to the blended sample, sample 11, the same kind of discrepancy, and sample 10, which is also a different percentage of the same kind of blended sample. We see, once again, a discrepancy and I'm just highlighting in these cases just some of the peaks that change and then some of the deviations we see. So what we were able to conclude about this is that there was some form of sample degradation over time. In this case, what you see is the methyl fingerprint of the spectra collected at 600 megahertz of one of the samples. And the blue is the initial data that was collected, which was the very first spectroscopic data collected on this sample, or the very first spectrometer used. And then we see in the red the data collected on the exact same sample after a year on the exact same spectrometer. And as you don't need to be an NMR spectroscopist to be able to see that there is distinct difference between the 2 samples. This tells us that there's some kind of change occurring, whether it's a change in higher order structure or some form of chemical modification that is occurring. So to flesh this out a little bit more, we collected some mass spectroscopy data on it and compared it to what we expect to actually see. So in the first column from the left, you see the title for NISMAB RM 8671, which is what samples 10, 11, and 12 are also, but at blended states. And you can see the relative amount of oxidation occurring at each of the specific methionines associated with that. As you move across the board, you can see that each of these samples has some oxidation occurring at different sites. So as you can see here, methionine 34 has a significant amount of oxidation compared to what you would expect to find. Methionine 101 has probably the highest level of oxidation compared to what you'd expect to find. And then methionine 255 also has, for the most part, some extensive oxidation as well, which goes to explain why we are seeing what is occurring in those spectra. So to just illustrate some of the outcomes that we've seen from this study, one, the profile method and the 2D fingerprinting method, for the most part, provide fairly comparable data in these instances that we showed, with the exception of being that the degraded samples were less sensitive or less able to be detected by the 1D method, or the 1D profile method, I should say, than the 2D fingerprinting method. And this could be due in part to the fact that when you add an extra dimension of detection, you can pick up on a few more things that you maybe won't necessarily be able to pick up on with 1D if it's a very low level of chemical modification. Now, if these were much more extensive, you would probably see that in profile quite well. So from that, I want to move on to some work that we've been doing that is very much related to this, using low resolution techniques of NMR as a screening technique for higher order structure. One of the issues that we run into as NMR spectroscopists is most people think that you need a very big, super expensive high field magnet, like a 900 megahertz instrument, which is going to run in the range of millions of dollars in order to acquire these kinds of spectral data. So we wanted to go and see if we could actually acquire good data, and if it was capable of giving us the information that we wanted at low field. So here we used 4 different samples, which I have listed, and we acquired the same kind of diffusion experiments that you would collect using profile, so the pulse-field gradient stimulated echo experiments. And we did this at 600 megahertz but with a cryoprobe, but we also did this at a lower field at 400 megahertz with a room temperature probe. And for those who don't know what I'm talking about as far as the differences, the lower in field strength you go, obviously the less the sensitivity, but you also lose a significant amount of sensitivity when you use a room temperature probe instead of a cryoprobe. And the reason we wanted to look at these specific setups is many companies have access to a 400 megahertz NMR with a standard probe. Much fewer, or many fewer companies have access to higher field instruments such as 6, 7, 8, and 900 megahertz NMRs with very expensive cryoplatform systems set up. So we collected this data, and what we were able to decipher is that using the profile method, so this is the 1D data that was processed using the profile correlation analysis, we see a similar trend across the board with these samples. The issue lies in the scaling of the difference of these samples, or the correlation similarity of these samples. So if you look on the left column, the 2 spectra, or the 2 plots you see on the left, are both at 600 megahertz with 1,000 scans. This data takes approximately an hour and 30 minutes to acquire. And the top and bottom are the exact same data points. I just organized it that way to illustrate a point. If you look on the right column, the 2 box plots, the top is at 256 scans using a 400 megahertz spectrometer, and the bottom is at 512 scans using a 400 megahertz spectrometer. The difference in time is at 512, using this setup, it takes approximately 40-ish minutes to acquire a spectrum. And at 256 scans, it takes approximately 20 minutes to acquire the spectrum. So at 256 scans, we can acquire essentially 5 spectra to get somewhat significant statistical values in roughly the time it takes to acquire 1 spectrum at 600 megahertz with 1,000 scans. Now, as I mentioned previously, the issue that we run into here is, while the trend is the same, if you look at the y-axis scales on these box plots, you'll notice that the 600 megahertz data only goes down to about 0.85 in the correlation value, while the 400 megahertz data goes down to around 0.5. So we do see a similar trend, but we don't see the statistical value that's being output being the same. And that does pose somewhat of a problem if we are trying to go across field strengths. So we are looking at a different method of trying to analyze this data so we can get somewhat statistically more meaningful data. So can we use principal component analysis to do this? Well, PCA, or principal component analysis, has been used on several occasions to analyze 2D NMR spectra, specifically the methyl fingerprinting method. And what I'm showing here is from a paper that was recently published in our group where they did a or an interlaboratory study and were able to use PCA to cluster the different sample types, regardless of the spectrometer that was used or the user. So the thought process was, well, at low field strength with low resolution, we're probably not going to be able to acquire 2D data, but can we do this kind of analysis for 1D data? So here we applied, or we tried to apply that thought process. So you see a series of 1D spectra that were collected. And does it work? Well, we think so, and we're still refining this. But we're able to acquire 1D data at low field. This is 400 megahertz with 512 scans. So each of these samples took about 40 minutes to acquire, or each. And as you can see, the clustering is very good from PCA. And not only is the clustering very good, but the variation between clusters, the distance between clusters is fairly pronounced, so we can actually see a difference. We're currently in the process of developing a method in order to determine whether or not what we're seeing is statistically significant. So that's still a work in progress. But at this point, it's very promising, and we're hoping to be able to push this out the door in the very near future. So just as a quick summary, to overview everything, 1D profile and 2D fingerprinting both provide comparable data as long as we are using a linear correlation algorithm. We are looking at developing PCA methods for 1D and seeing if that gives us similar information to what we would expect from 2D. We did see that 2D methyl fingerprinting is a little bit more sensitive to low levels of chemical modification than the profile method was, but we are also testing to see if the PCA method can give us a similar kind of output on the 1D data. We also found that low field, low resolution 1D spectroscopy methods are sufficient for screening at least, but more robust statistical analysis need to be implemented to make it really something that can be useful to the pharma industry. And then the 1D PCA method, we're actually developing into a toolkit in NMR pipe, and hopefully we'll be able to put that out the door to give other people access to the ability. And with that, I would like to thank and acknowledge people who are involved in this study and these studies. So Dr. Luke Arbogast at NIST, Dr. Mads Wikström at Amgen, Dr. John Moreno at NIST, Dr. Frank Delaglio at NIST, Dr. Robert Renson at NIST, and Drs. Yves Aubin and Human Gastriani at Health Canada, and then Drs. John Giddens and John Scheel also here at NIST, who provided us with some mass spectroscopy data. And with that, I would like to thank all of you for attending and listening.